So when we're talking about processing and integrating SAXS data collected in high throughput, yesterday I talked about this setup where we have 96 well plates, we're collecting a ton of data, and that presents a challenge for processing the data because, and we've relied heavily on farming the data processing out to users like Kevin and Chris, who have, handled their data and processed it. It's not like one sample or three samples like you get from, SEC, where, it's a tractable amount and you can, quickly get your SAXS results and then focus on the biology. If you've got a 96 well plate of results, it's impossible for Beamline staff to go through that on their own by hand. So we were working, relying on users to do that. And now we're collecting at a rate that even for users, it's becoming more and more challenging. So it not only presents a processing challenge, but also looking at the data together. I'm trying to advance my slide. And it's having fun with me. Side. All right, there we go. Let me skip that slide. All right, so I'm just going to, besides the wonderful studies that Kevin and Chris just talked about in terms of high throughput analysis, I'm just going to highlight 3 other examples that were published this year. So this was a study on nanodiscs, and I'm just describing the kinds of studies that you might think about doing with high throughput instead of, or in complement to SEC-SAXS. So this was a study of looking at nanodiscs and enriching the amount of a different type of lipid into the nanodiscs and seeing how that influenced nanodisc structure. So you can see if you have 100% POPG, it's a kind of a flat, narrow structure. As you introduce POPG, you start getting fatter and more elliptical nanodiscs. So you can imagine that being useful for certain membrane proteins and why certain mixtures work for some membrane proteins and not for others. But again, it's a combinatorial issue. Another example was looking at post-translational modifications. So there's a protein gamma crystalline in your cornea that is at 400 mg per mil and is transparent, but over time it gets apparently deaminated. And as it becomes deaminated, it starts to enter an aggregation-prone state, which causes cataracts. So 400 mg per mil is not common. So this group was looking at treatments of alpha crystalline as you progressively deaminate the protein and showing that in solution, different deamination treatments introduce the structure factor. So sometimes you would get repulsion between gamma crystallines and sometimes you'd get this aggregation that was not helpful. Another example, and one that generally hits tons of home runs, is Dave Baker's lab. I mean, they publish science papers a lot that utilize a lot of the SAXS Beamline, the high throughput. So in this particular study that was just published, they invented, it advanced without me telling so. They created basically the wheel and axle for proteins. There aren't many examples in biology. There's some the ATP-CAs in membranes, and maybe some the little pili that will rotate, but they're very rare to have wheels. And so the Baker lab was looking at engineering different types of protein, examples. They had to come up with something that assembles, that didn't slip off once it's assembled, and that was free rotating. And they created a whole bunch of these guys, which we hope will be used for some purposes. But in order to validate the structures, what you see is all the SAXS data on the right-hand side, the red versus black, and they're just using it to confirm that the design structure and just the pieces and the assembly were made. So it's a very simple way of using SAXS, and they've got like a protein synthesis team that's amazing, and of course their proteins are super stable. So this is the kind of plates that we would get for different types of high throughput experiments. If you're doing a construct screen like that Baker Lab, or like Kevin was talking about, and you're doing it in the same buffer, where orange is buffer, and you have your 3 concentrations that you'd measure. So this would be a plate you could set up for measuring that. You could double book your buffer on either end. So that would be high concentrations. We've been using the system for drug screening, pH and salt screening, and there you have one protein, same concentration, but you're varying the buffer. And again, we really recommend collecting buffer on either side of the sample, just to make sure that the error in subtraction isn't gone off. So you put buffers on either side, and then you have your drug mixture or your pH and salt screen somewhere in there. All right, so here are some controls in conducting a high throughput SAXS study. Before you run a control, before you send a full plate of samples, run some control samples first in high throughput. It's a lot of protein. So if you're talking 5 mg per mil, 30 microliters per sample, 48 wells a sample, you're into the 1.5 milliliters of that, or 7 mgs of protein. That's a lot of protein, and not a lot of you belong to that club, probably, where you can produce 7 mgs of protein. So make sure you've done your due diligence, run your control sample. You want to check for concentration dependence. For sure, run an SIBYLS sample. Also, the high throughput is going to not going to take care of your heterogeneities, right? So you're going to be running, if it's not a pure peak, knowing that there's heterogeneities in the sample. If you have a SEC, maybe it's a pure peak and you don't have to worry about it, but no, before you run it. I would include a control sample on every plate you send. So that control sample might be, your wild type protein every time. And, if you're super concerned about it, you could put it in each corner, in A2, buffer sample A2 would be wild type, and maybe H12, H11 would be your other wild type. Run a control on every plate. If you're doing drug screening, a lot of people will do it with DMSO, just to solubilize the drugs. But DMSO also can change the conformation of macromolecules on its own. So make sure you do whatever you're using to solubilize your protein, that you've done a DMSO screen, and don't just compare without, because we want to make sure that what we're studying is not DMSO effects. Please check for solubility, particularly with drugs. Sometimes, the drugs, even with DMSO, will come out of solution. And also maybe you've done an extreme pH, a pH of 1, and that aggregate your protein. So that's going to introduce some particulate matter, and that might, drift in and out of the beam as we collect, might cause some clogged needles. You might, at one point introduce some crud on the needle window that just sticks for the rest of the experiment. So at least be very aware of the solubility issues that you have. And if you know that there are solubility issues, please also let the Beamline scientists know that there might be solubility issues that we should look out for. There are some things that we're doing to help those of you who are not in the 7 milligrams of protein club. So Brandon's been working on very low volume needles. So Brandon claims that this 6 microliter in a well needle works. It picks up 4 microliters. Currently, we're asking for 30 microliters. And we're picking up 15. So this 6 microliter reduction is a 5-fold drop. So that puts you down to the 1 milligram of protein needed per plate, kind of. And I bet more of you belong to that club than the other one. So if we get this to work, I think it's going to be we're going to have a lot more projects. It's a big, a 5-fold drop in requirements is a big drop. So, a lot more projects will come. We're also working on quick exchange needles. So ones that currently, if we're running for a week, we'll probably have 2 needles for that week and maybe have 8 users or 10 users or something like that. So if we could quickly exchange needles, maybe per project, or at least someone's sending us some crud in the sample, we can quickly get out of it. It just allows for us to run more plates during the available time. Another thing that we're adding or have added, but not fully implemented, is UV on the side. So you can imagine adding in series with the collection. You go over the X-ray beam, you collect your SAXS data, then you go over to the UV area and collect the UV sample. So that's something that's particularly good for checking proteins with redox state issues or other things. So, this is implemented. We're now adding multiple plates on the deck, and we also have a DLS plate reader. So one thing that we could pretty easily do is pull up from your plate the plate that you sent, and then drop down into a DLS plate, so that you could both get your SAXS data and your DLS data and your UV data, in principle. So with the way that we're collecting data with this needle, we can deliver it to many different instruments that can accept that kind of thing. So if you have another technique that's interesting to add, and again, I'm, listening to the last couple talks, circular dichroism came into mind, or maybe some cross-linking, we might be able to apply those, but those are sort of long-term goals. And just to say that, we're using this plate reader, this UV cell, so that, we can look at carryover issues. So we can add something with color like blue dye into the needle, look at the UV trace, and then, look at the residual. This in particular was comparing first blue dye, then directly into water without a wash, and just seeing how much residual dye there is. So we're using this system a little bit now. I don't know that this movie will play. We will see. And I may have to drag over, and we'll see if I get kicked off the internet for doing this. And my internet is slow, so what I want you to see is that at the very top of the needle there, you can see the liquid level going up and down. So what you're seeing there is oscillation of sample during collection. So what we're hoping is that will greatly reduce, if not eliminate, radiation damage for most samples. And we've applied this, it works really, really well for everything we've tested. But occasionally we get a sample, and again, maybe Kevin was a victim of this, where you get a bubble or a low density amount of liquid in there. And if you look at the transmitted X-ray beam, the plug of sample comes in and out, and the bubble comes in and out as well, right? So you get these oscillations during your collection. So, this is something we wouldn't have added to the pipeline if it didn't work in our hands. But if you got something pretty sticky, or things aren't quite right, you might fall victim to some of this. So look out for it. Let me move this out of the way. All right. So try to advance here. See if I fall into the trap. Okay, great. So Chris already talked about efforts to reduce the challenge in processing data. So if you're getting 48 samples in a plate and you've sent us 3 plates, that's a lot of data to crunch through. Probably too much, that's going to take you maybe a month. So what we would like to do is we would like to help process the data for you. So I'm going to go through the frame slice tool in a second. But this is how the radius gyration changes as the number of frames. So that's a really good indicator. And these are two different samples and how that behaves. So we have tools. Chris has developed the tool. Other people are developing tool, and we're trying to add that to the pipeline so that you get whether your buffer 1 subtraction agree with your buffer 2 subtraction, whether it's aggregated or not, what the RG is, and then how many frames you can take. So the number of files that the RG region stayed the same, given buffer 1, given buffer 2, and the average. So these kinds of things. So yeah. So with that, I'll pause here for a second, and I was going to go into a practicum. Mikhail, how much time do I have again? Catherine?
Sorry, hang on.
Yeah.
45 minutes, I think, but let me check.
That was probably the whole talk. But anyway, are there questions? I was going to jump into, using the frame slice. All right. So let me, let's see. Stop share here. And now I'm going to share over here of results here. And you should be able to download those to your own terminal through RCP or whatever tool that you use to pull data down. So I've done that already. I want to escape out of presentation mode. Yeah, there we go. Workshop. So I've pulled data down. So avgraphs. The avgraphs file contains a summary of the data, and you can scan through this. This is your buffer 1, buffer 2 comparison. Gives you a sense of how much radiation damage you have, and you can open it in whatever way you want and just kind of scream through that. Sorry, I'm probably moving faster than Zoom can handle it. To get a sense of that. Let's see. I'm going to close that. And yeah, and the set I'm going to analyze here is going to be a GFP set where I'm varying which salt is in, from cesium chloride to lithium chloride, and the concentration. And this is for examining the hydration layer. So I'm going to quickly just kind of go through that. So this is the GFP. It's a very strongly charged one. Looking at that plate, I'm going to pick something pretty, to begin with, pretty standard. So let's look at the 100 millimolar sodium chloride. That would be what you typically send, a typical concentration of sodium chloride. So that's in C2, 3, and 4. That's going to be particularly nice. Let's see, I want to look at this folder. So I'm going to look at the results, the subtracted results, and I want to look at C3. And you've got your buffer 1 subtracted from the sample, your buffer 2 subtracted from the sample, and the average. You should be able to go to our Beamline. It has all our web applications. So you can go to web applications, and you can go through the frame slice tool. And then you should be able to drag and drop, the buffer 1 in here. And that should load. And you should be able to, so what you're seeing here is the very first frame collected, all 33 frames analyzed together, and you should be able to see whether there's radiation damage. And you can look, and we separated this into 3 regions. So you can look at the Gunier region and just see how many frames you can include before radiation damage occurs. And there's a slight amount of radiation damage in this sample. So you can see as I'm increasing the number of frames, the teal curve, which is 7 frames averaged together has drifted a little bit away from the very first frame, but certainly below the average, all of them averaged together. So there's some tiny bit of radiation damage here. You probably wouldn't have noticed it. And it probably doesn't make a huge amount of difference in terms of the analysis. You know, it's probably a half angstrom difference in radius of gyration. But here I'm examining the same protein, just in different salts, and I'm really looking at the hydration layer and whether, you know, cesium comes onto the surface of the protein or not. So these are exactly the things that I really want to pay attention to. So these half angstrom differences are important. So I can, you know, so basically that's what you're looking for. In the data set, we've already started including a file here, an RG analysis results file. It's in the RG stats, and it's not particularly reader friendly. Like, let's see, I'm trying to open with. I might have to click on it and open with Notepad. I used to have a version that was HTML friendly like the other screen, but somebody updated Python and that broke. But so looking at C2 buffer 1, it's saying that you can use the first 11 frames before the radius of gyration changes significantly. So, and that was kind of where we were at. I was going at 7, saying like somewhere around there is where the noise from the first frame is high enough that you can accept up to 11 frames and still be within the noise of the first. So it's doing some kind of, not some kind, I wrote the code, so I know exactly what it's doing. But it's doing an analysis of the files. So again, you would do this for all of them. So that's just gives you a flavor. You would download that, and you would then you know, compare buffer 1 and buffer 2 against each other to make sure they agree, and pick one of those two. So you again assess those. So that's kind of the workflow of processing 30 frames. Again, I'm looking at a whole plate worth of this, right? So it takes some time. And as those of you who have done this know, it can be really numbing with all the data that we're producing. So again, that's why we're trying to create automated scripts for it. Let's see. I think that's all I wanted to do there with that. Go back to my talk here and mention that, I guess I'll just go this way. So we have a new repository for correlated data sets like the ones that we're generating. It's called Simple Scattering. And it is meant to be not only a repository for correlated scattering data sets, but also a place where we're going to be starting to store all of your SIBYLS data sets, or we're thinking about it. We're actually prototyping it now. It may work. I'm hopeful it'll work, but we're going to ask for community involvement in helping us develop that further. So going to the web apps, going to SAXS sample server. There you go. Nope, not sample server. That's where you deposit your spreadsheets. I want, sorry, it's underneath the Zoom bar. Simple SAXS, there we go. And I can type in here, you know, GFP. And this is a deposited data set. It has a code associated, much like a PDB code when it was created. And this is green fluorescent protein supercharged with mining 29 with the chloride salts. And I deposited this, and I have a description and, you know, where it would be published and the design. We're adding information about the sequence and the source of the material. But we would like you guys to deposit your high throughput data sets here. We think that tools are very necessary to develop for analyzing such correlated data sets. I think, you know, people who are developing the SEC-SAXS code would only benefit from having data sets that are SEC-SAXS. So if I go to all data sets here, you know, there's some SEC-SAXS data sets here, and this is the full spectrum. So for tool building, this is what we're hoping for. So I'm going to go to GFP, view it. I'm going to pull this guy down. All right, and I'm going to show in folder. So this is the full data set. I'm going to open it. It's a zip file. Let's see, extract all, extract. And inside are the data sets of the data. So that's deposited there. Yeah, I'm gonna now talk about using similarity maps to analyze such data, okay? So also at the Beamline are SAXS similarity maps here. So I'm going to illustrate what this is. There's a tutorial in here by using an example data set that's available there. So this is a cage, and this is theoretical data of this cage in multiple conformations going from a open state, like the one shown here, to a closed state. So it's a linear morph between the two. And you can pull that down using that set, that morph.zip there. And then you can drag and drop. Let's see, I want to look at, not the downloads, because that's what I just pulled up. Workshop, and it's not there. So I may, let's see, I must have put it down, pulled it down on downloads just a second ago. Oh yeah. So here's the morph. So the way that I've deposited that data set was in a random order. So the second frame parts, that is the steps in the morph. But I've randomized them here so that when you pull them up, they'd be named. And if you label them alphabetically, it's going to be randomized. So I'm going to dump all these SAXS profiles into the SAXS similarity map. So this, what this thing does is it compares each cell as a comparison of two SAXS profiles. So if I click on this one, these are the two scattering profiles that are compared. So this is randomized frame order 0, 0 against randomized frame 0, 1. These are actually step 2 in the morph and step 16 in the morph. So it's comparing how similar these two curves are to one another. The number reported here is the difference in raised duration. So you can show RGs, and it'll show all the differences in the raised duration of all of these frames. Because it's randomized, you know, you're going to have to sort through it, but there is a clustering function. You can cluster it, and it'll cluster it based on how similar, or the reddest things will appear close together, and the ones that are far apart will appear far apart. I'm going to hit cluster here, and what you should see is that based on how similar the SAXS profiles are to one another, it's self-organized by frame number. So the steps in the morph, right? So frame 0, the very start of the morph, which is the open cage, is now right next to the first step. So it's using, it doesn't know anything about the structure. All it has is a SAXS curve to work with, right? So it's just using how similar the SAXS curve is, and therefore defining how similar the structure is. It's an inferred thing, right? And so it pretty much gets it all right. I guess 19 and 20 are pretty flipped. I can switch them over if I want to by hand, but that's about right. So that works. There's another option for visualizing such a data set, and that's using this force plot option. And it's pretty trippy, so I apologize for it. But basically what happens is each of the SAXS curves gets displayed as a node, and the size of the node is going to be proportional to the raised duration. So the closed cage is over here, and it has a force that's correlated to how similar they are. So things that are very close in similarity will be displayed next to each other, and it'll go around, and sometimes when things are very close, there's lots of false minima, so you kind of have to shake the structure a bit just to de-knot things. But this, again, it could be useful to you for analyzing the data. So I'm going to just go back to my talk and just say, this is the steps in the morph that I just talked about. And I've in the example, I used 20. This is just 11 steps. These are the SAXS curves from each of these steps. If I drop them in randomly, you get something random. But if I cluster them in a sensible way, in a sensible algorithm, that's what I mean, you get something that's pretty meaningful to you. It's how similar things are. As was mentioned by Kevin before, you can do things by chi-squared, and that's what most people would do. We've created a different metric for quantifying how similar two curves are. It's called the volatility ratio. It basically takes the ratio of two curves and sees how volatile it is, just like stocks. And that correlates much better with this the RMSD differences. So as you step through this morph, it's a linear morph, right? So the RMSD difference between this structure and this structure should be about the same as between this structure and that structure, etc. And so the V sub R seems to track much better with RMSD. Chi-square just loses all capacity to do so. It's basically a binary measure by the time you get to the third step. So it says yes, similar, or no, not similar. So you oscillate between 1 and then 10, 15, and it's just not similar anymore. So we really like this V sub R. It's not the end all. There's lots of problems with it, but I like to use it for this purpose. And so just joining those two tools, simple SAXS, you got your correlated data set, you can pull that down. You can go then to your similarity tool, so here. And you can, let me see if I can find that data set now. Workshop, GFP data. Look at my results folder. Oh no, wait, this is the original. I want from the website. Sorry, I'm confusing myself here. Let's just do it again. Go to, not that one, not that one. I'm looking for, let's just go straight. Simple SAXS, GFP, view, pull this down, showing folder, extract all, extract. All right, so got my salt concentration. I can go to similarity map. There you are. Find that window again. There it is. And I can drag and drop that to assess how similar these curves are. And you can see that one of them didn't pop up. There's an X here. And it's the rubidium chloride. They're still here. It's hidden under the name. But at least it's well, the X is covering things, but 100 millimolar rubidium chloride didn't pull up. So I can look at that file and just see why things didn't like it. So I can go to rubidium chloride. Sometimes they won't pop up, and it's usually something with the some header or something. I don't know about that. Anyway, you can examine it. I'm gonna not do it on the fly. Let's see, it was 100 millimolar rubidium chloride. Anyway, here's the list, 500. Anyway, here is the similarity plot. Sometimes there's noise in the data, so sometimes you want to, and I just picked the one that disagreed the most, right? And immediately I can see that that's 500 millimolar cesium chloride and 500 millimolar lithium chloride. So that just says something, that those things are very different scattering curves, and here are how they're different. And sometimes the asymmetry here means you just have to clip the data around. You can adjust the Q range here, how many Q spots you want to use. The Q range may vary, but you want to kind of change it so that it's symmetric around the diagonal. Because again, this is comparison of curve 1 versus 2, and this is curve 2 against 1, so it's a symmetric matrix. But again, you can pick out what's really, really different. Sometimes you have something like this, where there's one curve like cesium chloride that's really different from everybody else. It's a bright yellow streak across the whole thing. And it would be great if we had a little contrast bar here, but I don't have that. Typically, if I'm trying to develop insight into a data set, I might not load the 500 millimolar again. And it looks like, the 100 millimolar cesium chloride is also a problem, right? So, that's that. This has been loaded in, not in a sensible way, but I can shift things around. So for example, I could take all the lithium chlorides, that's 10 millimolar, 100 millimolar, and 500 millimolar, and pull them down. Because of this big contrast here, where this is setting the extreme, these all look very similar. But in fact, when I kind of look at it in detail, there are differences here that it just can't pick up by because of this very extreme one. So I can pull that out and then look at the trends that I'm observing. Again, I'm looking at all the data here all at once. So that's the goal of this tool. I'm going to go through some examples. Catherine, you said, what time? I didn't pay attention. I was too nervous.
We are 15 minutes over. Great.
Well, I was following what Catherine said, so she gave me till I think 2:30. So examples of what you want to do. This is a protein called mutas beta. It interacts with DNA, ATP, ADP, so you can create heat maps based on the analogs that are available to you with and without DNA. You can develop insights into similarities. And these are curves that, are quite subtly different from one another. And, if, we measured, I think the maximum concentration I could get this guy up to was like 2-3 mg per mil before it starts to have problems. So, these subtle differences you may not be able to pick up in SEC-SAXS. So it really is a high throughput project. This is this cage again, and we're looking at different salts and pHs and throwing all the different, so this is 10 millimolar sodium chloride, pH 6, and this is 500 millimolar sodium chloride, pH 8, looking at this cage and plotting all the data. You can see this sort of grid-like pattern here, so you can examine that. Where this occurs is in pH 10 and 11, pH 10 and 11, pH 10 and 11. These are the big extremes. So you can examine that. We hunted that down, realized that it is because this cage is disassembling and forming trimers. If we look keep it in a happy pH, pH 7, and then look as we increase our salt concentration, we can see that as you go to low salt, you're closer to a compact cage. This is the sort of moving compact cage. And then as you increase your salt, you're expanding it to the open cage. There's just a gradient of similarity as you go. So salt is a way for opening and closing the cage, and pH is a way for disassembly and assembly. So there's an example there. It's probably too complicated to explain this one, but for engineering, you can use it. You can do, so Baker Lab was doing helix turn helix motifs, and they were examining all their constructs. So you can plot, your experimental data on one axis, your calculated data to see if their algorithm was working. So you should get a diagonal similarity if it is working. So it sort of worked in that case. And I think I've run out of time, so I won't go through the remaining bit, but hopefully you see it's a useful tool and at least a step in the direction for analyzing a large number of data sets, whether you've generated them in high throughput or in SEC. Hopefully it's useful to you. So I'll stop there and see if there's questions.